Can you hear me? Can you
yeah,
see my slide?
Thanks. Yeah.
Perfect. Thank you, Fuzhou and other organizers for giving me opportunity to participate in this such a wonderful feat. I think this conference is reaching more audience than face-to-face conferences, so I begin to love it. I'd like to continue the discussion of epigenome mapping in single cells, continue what Joe beautifully started, and discuss the atlas of gene regulatory elements in adult mouse cerebrum. Here's the motivation. If you look at all the disease, neuropsychiatric disorder are in fact the leading cause of burden measured in life years lost due to the illness in the US. And it's the top 3 globally for disease burden. So it's actually a major health risk. And there's very few cures for neuropsychiatric disorders. Think about addiction, think about Alzheimer's disease. We are, as a society, facing tremendous challenges in coming up with cures to such such disastrous disease. Genetics can help. Genome-wide association analysis have uncovered numerous gene loci that are risk for late onset Alzheimer's disease, and as Joe also mentioned, this schizophrenia and other disorders. However, the major challenge still we face is how to interpret these variants, because most of them are in the non-coding parts of the DNA, and they're not well annotated in terms of their function. So a major hypothesis that has been proposed over the years is that they function by perturbing enhancers, distal regulatory elements that act remotely to target gene to disrupt transcription in specific cell types. To test such hypotheses and to really advance our knowledge of genetic mechanisms of neuropsychiatric disorder, we face a couple of bottlenecks. First, what are the cis regulatory elements in the genome that are affected by risk variants, and in which cell type are they active in? And secondly, what genes are targeted by such variants in the susceptible cell types? These are major hurdles that we biologists face, and how do we address them? The key to address this is to look at the genome, not as the individual sequences, but as a blueprint that actually are regulated by an epigenome in different cells. If you look at the brain, the human brain consists of more than 100 billion cells. Each of them contain the same genome, but they have different epigenome, as Joe nicely illustrated. The epigenome includes DNA methylation, exemplified by the CG methylation. They also include other modifications, such as histone tail modifications. There are hundreds of such modifications now. Also include higher order chromatin structures, as we heard earlier through Peter Fisher's talk. And also there are open chromatin that are considered part of this collection of the epigenome. The output of the genome in each cell is, in fact, regulated by the epigenome. For example, DNA methylation controls the accessibility of the DNA by transcription factors. Similarly, open chromatin also provides such access. Histone modifications regulate the openness of the chromatin as well as the compactness of the chromatin and the three-dimensional structure. So understanding the genome sequence is inseparable from understanding the epigenome in different cell types. Let me start with a targ-seq. You heard about this already. In 2013, Will Greenleaf and Howard Chen at Stanford University invented the method called assay for transposes accessible chromatin. They utilized a transposes in the bacteriophage transposon 5, co-opted into a double-strand DNA cutter. So it cut the DNA simultaneously, ligate a pair of DNA oligo to the ends of the DNA. That in one step create a single short DNA fragment that can be amplified by PCR. So in this simple and effective assay, you can start with small number of cells and map the open chromatin in the DNA. A few years later, single-cell targ-seq was born in 2 labs, one by Will Greenleaf, another by Jay Shenjoy, who talked earlier. And they, in Jay's lab, they invented a strategy that they call single-cell combinatorial indexing, a targ-seq. In 2 steps, they could add combinations of DNA barcodes to the open chromatin DNA fragment generated by the transposes, and in the same time creating tens of thousands of combinations of barcode that allow them to identify open chromatin in a single file fashion. So we mainly, Sebastian Prezo, a former postdoctoral fellow in my lab, adopted this strategy and optimized it for fresh and flash-frozen biopsy samples. Further, he started a platform in the UCSD called Center for Epigenomics, where he successfully used automated system to carry out this whole assay. So in a very short time, we could profile actually up to 40,000 cells per experiment with average of 5,000 open chromatin fragment assay per cell. This is some examples where individual cell, you can see hits of the open chromatin, and when you pile up everything, you can see in the population sharp peaks accumulating in the right locations. There is a problem though, that data is still sparse. 5,000 fragment sounds a lot, but it's only about 5 to 10% of the open chromatin that we estimate to exist in each cell. How do we deal with that? My student, Rongxin Feng, former student, come up with a strategy to process single-cell targ-seq. He called it snap attack. It's now quite popular in the field now. What this does is after pre-processing, he would treat each cell using a string of 1 and 0s, with 1 represent open chromatin, 0 represent inactive part of the DNA. Then he would assay and calculate the similarity between each pair of cells to build a pairwise similarity matrix. Through this similarity matrix, he can then group the cells that belong to the same group together, here represented using a T-SNP, but now we use UMAP to do it. After that, we can now use individual, use cells from aggregate the profiles from each cluster into aggregate profiles, and you can basically identify cluster-specific peaks. You can annotate the cell clusters based on the open chromatin at marker genes, and use that to define cell types and link to the transcriptome profiles. So I will show how we use this strategy to gain a better understanding of the regulatory landscape of mouse brain. As Joe mentioned, we're part of the Brain Initiative Cell Census and Circuits Network. So in the first few years of this consortium, Joe and my lab, with the help of Margar, profiled a single cell resolution, both chromatin accessibility and the DNA methylomes, coming from cells from the cerebrum, which consist of isocortex, olfactory bulb, and hypothalamus and the subcortical nuclear areas. This is the largest parts of the mammalian brain, and they're responsible for motion, for learning and the memory and the space navigation, and for smell. So this is obviously the central nervous system that we care about. So through a taxic analysis of about 800,000 nuclei coming out of 45 brain regions that are dissected by MAGAR by hand over the course of the last few years, we managed to then group and identify 43 major cell types in this parts of the mouse brain. And this was shown here. And upon further subclustering, we can identify a total of 160 subtypes. And they were matched quite nicely with what Joe identified using a single cell methylome. Again, we found very consistent cell typing. This is remarkable because now you can use two different molecular modality to identify essentially the same subtypes, suggesting that this the different cell types indeed exist and can be separated based on their unique epigenome profiles. This slide shows how we annotate each cell clusters. As I mentioned, we used open chromatin profiles of major marker genes of each cluster. And these marker genes were known from the literature and also were identified from single-cell transcriptomics maps. So this gave us one level of confidence that our mapping results are reliable. We also perform unbiased alignment of single-cell taxic data with single-cell transcriptome data that was published a few years ago from Stan Linnerson's group. And we can see almost a very good alignment between the cell types identified by the transcriptomic data and by accessibility. So E type represent accessible chromatin based chromatin cell clustering, and T represent transcriptome. And you can basically see this in the diagonal line, suggesting the 1-to-1 or 1-to-2 or 3 correspondence between each modalities. In fact, out of 160 cell types that we defined, we can find corresponding cell types defined by transcriptomics for 155, and vice versa. From 100 transcriptome defined cell type, we were able to find 84 of them using the chromatin landscapes. Here is a website I like to invite you to explore. Remember this, catalyst.org, mouse brain. You can basically explore both the accessible regions for each of the 160 cell types that we annotated, and also correlated accessible regions as exemplified by these arcs. Out of these 160 cell types, for each one of them, we were able to identify peaks, and altogether we identified 491,000 open chromatin regions in these cell types, and they show a strong cell type specificity as shown here. Cluster 1 are mostly ubiquitously accessible, but other clusters show extreme variabilities among different cell types. We were able to connect many of those regulatory elements or candidate regulatory element with their target genes through correlative analysis. So first we identify co-accessible pairs using the single-cell taxi data, and then we compute the Pearson correlation between the accessibility of those regions across cell types and gene expression of the nearby genes. And that gave us this correlation. And you can basically see that for 129,000 pairs of distal element and genes, we can see strong positive correlation, suggesting potential involvement of such element in the gene expression. And these are listed here. You can basically see that there's a large number of pan-neuronal elements, and they are accessible in the neurons and less so in the non-neuronal cells. And transcription of linked genes are showing neuronal gene-specific neuronal transcription and absent of transcription in non-neuronal cells. You can also find many other cell type specific regulatory elements that are showing correlated gene expression in the corresponding cell types. If you look at the open chromatin of these promoters, the promoter accessibility of these regions are largely invariant, suggesting that a distal element is the driver of the cell type specific gene expression. We can perform motif analysis, identify cell type, identify transcription regulators that are likely involved in regulation of different sets of genes. And we were able to identify 235 such transcription factor motifs, making predictions for major cell types in the brain. The question that we wonder is, do these elements show conservation in the human? And if so, do the homologous regions in the human genome show enrichment for neuropsychiatric trait-associated SNP? We found that out of the 491,000 elements that occupy roughly 14% of the mouse genome, roughly 70% can find a human homolog based on homolog search, one-to-one correspondence. And we assume that those human homolog sequences potentially could act as regulatory elements in the human corresponding cells. So with this assumption, then we perform the enrichment analysis, looking at the enrichment of different neural trait associated sequence variants in the homologous sequences of mouse brain regulatory element. And we found, not surprisingly, very strong association of schizophrenia with regulatory elements that were uncovered in the excitatory neurons and inhibitory neurons, and also educational attainment associated sequence variants to be also enriched in subclasses of excitatory neurons, such as IT layer 4, global ITL4GL neurons, and at some other inhibitory neurons. Bipolar disorder, another neuropsychiatric disorder, is found to be associated with largely inhibitory neuron enhancers. So this is a vast data set that is now, for the first time, provide a landscape of transcriptional regulatory elements for exploring mammalian brain cell types. And you can explore them from the website catalyst.org. Now, there still are more questions. First of all, detecting open chromatin is just part of the story. What is the activity state of such candidate regulatory elements in different cell types? That's still unknown. And how do we define the targets of these regulatory elements with better defined elements? So besides open chromatin, another indicator of activity state is histone modification. As shown here, histone tails can be modified, either acetylated or methylated, and acetylation indicate active states of the chromatin is typically found at active enhancers or promoters. Methylated histones can be both active if it's found in some lysines or repressive if it's found in other places. And histone is known to, modifications known to be recognized by reader proteins that perform additional functions. To identify targetings, we also heard today that three-dimensional chromatin context is important. And that can be measured by methyl high C, for example, that by high C or single cell methyl 3C, as Joe just mentioned. And that provide a map of chromatin contacts. Why we are doing this? I think this is particularly motivated by a recent report from Echtlander and Jesse Grace from Broad and Stanford University, where they developed a model called activity by contact that fairly accurately predict enhancer targets. The idea is very simple, in fact. After you identify open chromatin, you can use lysine 27 acetylation state to estimate the function or activity of that distal element. But that alone is insufficient to predict targets if you just look at the nearby gene. In fact, you also need the contact information provided by the high C experiments. So if you multiply the histone acetylation state with high C contacts, you can generate a score called ABC score that fairly accurately predict how much enhancer contribute to a target gene expression. So this was published last year in Nature Genetics, and in our hands, in others, has performed quite well. But how do you measure chromatin state at single cell level? And how do you actually group cells together from the same cell clusters? That is not trivial. And there have been a lot of attempts to this. And my postdoctoral fellow, Chen-Xu Zhu, who was in fact a trainee at Chen Qi's lab at Peking University, developed a method called paired tag that allow us to analyze histone marks and transcription jointly from the same cell in a very high throughput manner. This is how it works. The first step is taken from a cut and tag method that invented by Steve Haneka from from Hutchinson Cancer Institute. So in this step, you stain cells with antibodies, and then following that, you can use protein A fused to transposase 5, TN5 transposase, to introduce double-strand breaks and DNA adapters at locations where the histone mark and antibody associate with. But at this step, nuclei are still intact. They're just lysed. And the DNA that are cut was kept in place. And following, actually, it's not yet cut here, but it's just bound by the transposase. Following that, then you can perform the single cell combinatorial indexing as Jay introduced earlier. In this step, you could use multiple rounds of ligation to add DNA barcode to the open chromatin or to the reverse transcriptase made by, to the cDNA made by the reverse transcription reaction. So we can perform this multiple rounds. In fact, we can do this 2 or 3 rounds to create millions of combinations of DNA barcodes. Afterwards, you can basically split the DNA into 2 parts, one for DNA library, one for RNA library, and you can basically identify the DNA and the RNA component of each cell. To demonstrate the utility of this strategy, Chen Xu applied it to frontal cortex and hippocampus from adult mouse brains. For each parts, he performed the joint RNA and histone modification analysis, focusing on 4 different histone marks: lysine 4 monomethylation, an active enhancer mark, lysine 27 acylation, also an active enhancer mark, lysine 27 trimethylation, a repressive mark, and lysine 9 trimethylation, another repressive mark. And then, because each cell contains the RNA component, we can basically align the cells based on the RNA profiles, and then ask what the histone modification look like. So basically, out of 45,000 nuclei profiled, we identified 22 clusters. They are listed here, corresponding to hippocampal neurons, cortical neurons, multiple classes of inhibitory neurons, and many different kinds of glial cells. And this is what you see when you aggregate cells from the same cluster. You basically see in the neuronal cells, marker gene SNAP25 show very strong RNA expression and show histone modification profiles and show lysine 27 acylation at the promoter and the distal element, and the lack of lysine 27 trimethylation, but presence of this repressive mark in the non-neuronal cells. Similarly, neuron D is a neuronal mark, excitatory neuron mark, and you basically can see it's transcribed in the neurons, but not in the inhibitory neurons or non-neurons. And you basically can see corresponding active chromatin marks of lysine 27 acylation around this gene locus, and a lysine 27 repressive mark in non-neuronal cells or inhibitory neurons at this gene locus, and so on. This is a slide to show why you need to do joint analysis. Basically, you can cluster cells, also use histone modification clusters, and that will give you somewhat different result. If you use histone H3 lysine 4 monomethylation to cluster the cells, the resulting cell clusters match fairly well with the cell cluster that you make using RNA component. Similarly, if you cluster cells using the lysine 27 acylation, the two approach give you similar readout. However, lysine 27 trimethylation mark give you poorer resolution. In fact, it could not resolve all the cortical neurons. They all look the same. Even worse, lysine 9 trimethylation does not resolve subtypes of neurons at all. They all look almost the same. So this illustrates how important it is to have the RNA component in this clustering approach. Without RNA cluster component, it's virtually impossible for you to link different histone marks together. With this map, which was generated for the first time for mouse brains, different subtypes of the mouse brain, we can then examine the chromatin state at both promoters and distal element in different cell types. So each column here represents a cell type, major cell type that we identified using RNA-seq analysis, and each row represents a promoter here. Essentially, you can see that a large number of promoters are silenced by lysine 9 trimethylation in the neurons. And what they are involved in signal transduction, sensory perception. They are, in fact, olfactory neurons that are genes that are largely silenced by this mechanism. There's another group that are corresponding to promoters that are silenced by lysine 27 methylation, and they correspond largely to developmental genes involved in neurogenesis and other developmental processes. And they are largely silenced in neurons and non-neuronal cells. And then the remainder promoters that are active in the neurons or non-neuronal cells, they are associated with lysine 27 or lysine, acylation or lysine for monomethylation in a cell type specific manner, and they correspond to different subtypes of neuronal specific genes. This is the chromatin state at distal element, and you can also see a large number of distal elements to be regulated or to be at least occupied by lysine 9 trimethylation or lysine 27 trimethylation in a cell type dependent manner, and also a large group of distal element to be associated with active chromatin marks also in a cell type dependent manner. And you can perform motif analysis to identify candidate regulatory elements, transcription regulators targeting such element. Again, we can perform correlative analysis linking the chromatin state at such regulatory elements and their putative target genes. And this basically shows that acylation of lysine 27 is associated with a large group of genes in terms of possibly in transcription, and trimethylation of lysine 27 is associated with a large number of genes negatively across these cell types. And these are the elements I just mentioned that shows positive correlation between acylation and negative correlation between trimethylation. There is a half of these genes here identified as negatively regulated between lysine 27. Trimethylation is also found to be positively regulated, associated between acylation, which is expected. So in the remaining time, I just quickly go through this. You already heard from Joe that you can use single cell combined multi-omics, examining both Methylome and High-C. There is around the same time, my group, mainly led by Jason Lee or Guoqiang, who is going to join Beijing University in very soon.
One more minute.
Yep. So this is a single cell methyl High-C achieve the same goal as single nuclear MEMC-seq. And basically, as Joe said, you can use Methylome to group cells together and then use the aggregate contact information to identify chromatin contacts. So this is the last data slides. I just want to mention that with these two multi-omic tools, we actually can now make reasonably good guesses about what target genes for each cis-regulatory elements can actually be targeting and interpret the variants in each element. Finally, perspective into the future, I think human brain is a fascinating organ to explore, not only because its involvement in human health, but also scientifically it presents a lot of challenges. And we have to thank a lot of pioneers in this field. Paul Allen, who recently passed away, was instrumental in launching this effort. Started with the Allen Brain Institute and now jump-started into the Brain Initiative. This is acknowledgement slides. I already mentioned the key contributors of this work I presented and my collaborators, Joe, Maga, Minghu, and Yaping. And I'll stop here, take questions. Thank you so much for your attention.
Okay, thanks, Bing, for the exciting talk. So any questions?
Okay, hi there.
Hello.
Okay, I have a general or maybe biological question. We know in the brain, the cell types can be very different across the area and brain regions. And in your data, I see some like OB GABA or TI neuron maybe. So how do you determine where that the specific type of a certain region is really specificity of the brain region, but not from the sample bias. Given that you have 45 regions, and maybe each region is so big, not that accurate, and you just maybe have 2 replicates, right?
That's a good question. I didn't have time to talk about it, but each brain slice is about 600, about half a millimeter thick, and was hand carved out, very small quantity by Marga. So she basically take the Allen brain atlas, and basically from that an atomic map carve out the brain region. So we indeed perform replicate for each brain region, and we double check to make sure that the cell type composition and cell identity from the 2 replicates are consistent with each other. So that's the quality control that I didn't have time to talk about. Secondly, about brain region specificity, I didn't have time to go over, but it's true. Some neurons are highly brain region specific. This corresponds to cortical excitatory neurons. Other neurons are more or less uniformly distributed. These are non-neuron cells or inhibitory neurons. So major classes of inhibitory neurons, you can find them throughout most brain regions. This is all described before, so I didn't talk about them.
Thank you.
Okay, Yicheng, please.
Thank you for your talk. I have a question that the K27 trimethylation and the K9 trimethylation are both suppress histone markers. So why they suppress different genes? What are the differences between these 2 histone markers?
That's a very good question. Lysine 27 trimethylation is catalyzed by so-called polycomb repressive pathway, and they are read by PRC1, so polycomb repressive complex 1. They go through a different mechanism in terms of their catalysis. They are known to regulate mainly developmental regulator genes. So they are probably evolutionarily evolved as a distinct mechanism as the lysine 9 trimethylation, which is also known as heterochromatin mark. Heterochromatin, as you know, is implicated in nuclear membrane mediated silencing or nucleolus mediated silencing. So they are mainly catalyzed by different sets of machinery, and read also by different set of machinery. So the genes that are silenced by heterochromatin appear to be mostly repetitive DNA in nature, such as olfactory receptor genes. These are duplicated many times in the human genome or in the mammalian genome. So such repetitive DNA sequences is known to be silenced through lysine 9 trimethylation pathways.
Thank you.
Okay, Bing, I have a question. So you mentioned that for the H3K9 trimethylation is very similar between different type of neurons. But after you using the RNA to separate the different neuron types, do you still see some subtle difference between these neuron types of the K9?
It's a very good question. We haven't. We actually have a slides. So we do see subtle differences between different neural types once we separate the cell types. So we can identify cell type specific lysine 9 trimethylation or subtypes according to different neural classes, especially at the so-called distal element.
Okay. Okay, so if there's no more questions, let's thank Bing for the exciting talk. And also let's thank all the speakers this morning.